few set of on caffeinated smoke as linked to type 2 diabetes and obesity, data from the National Health and Nutrition Examination Survey. So that needs to be way up there. This one up. Maybe a little higher even up there.
Oh, there we go. Yep.
Got it.
Good. Okay.
Yeah, that one.
So can I? Good morning. Today I'm going to talk about secondhand smoke, its relationship with type 2 diabetes and obesity. This is being done by myself, Dr. Friedman, Dr. Shaheen, and Mr. Pham. It is a chronic disease that occurs either when the pancreas does not produce insulin or when enough insulin is not being produced. Insulin is a hormone that regulates our blood sugar. There are two main kinds of diabetes. There's a third one, which is gestational diabetes, but I'm not going to focus on that. Type 1 diabetes is when the body does not produce insulin, and type 2 diabetes results from the body's ineffective use of insulin. It is the main kind of diabetes. In the United States, 25.6 million, which is about 11% of all adults aged 20 years and older, have diabetes, according to the CDC. Over the past 20 years, there has been a drastic increase in the rate of obesity and overweight in the United States. According to the CDC again, about more than one-third of the U.S. adults are obese and more than one-third are overweight. Cigarette smoking is the leading avoidable cause of death in the United States and is the 7th leading cause of death in the U.S. About 400,000 in the U.S. is caused by cigarette smoking. Nicotine is a metabolite found in nicotine. It's found in cigarette smoke. In this study, we're going to focus more on nicotine. There have been various studies on the relationship between cigarette smoking and the rate of type 2 diabetes. But it's surprising because we know that smokers are more than non-smokers, whereas obesity is the rate of diabetes. However, the relationship between secondhand smoke and diabetes is less established, and that's why we are starting that research right now. The main objective of this study is to look at the relationship between nicotine level of verified secondhand smoke and diabetes, and also overweight and obesity. And when we talk about nicotine level verified secondhand smoke, we mean we had a questionnaire that asked people whether you smoke or not, and people said no, whereas they found nicotine in their blood levels. We use data from the National Health and Nutrition Examination Survey and HANES is a national survey conducted by the CDC, and it's across the states. It's based on all the U.S. It is on non-institutionalized population of the United States, and it has data from babies all the way up to about 89 years of age. It has data on lab, examination questionnaire, dietary, and even genetic data. In this study, we're going to focus on years from 2001 to 2006. And Hayes was in 1976, and it goes all the way up to 2011, and it still continues. Our study population was on 6,219 adults aged 20 and older with data on our outcome variables. For this study, we focused on obesity and overweight, looking at BMI, and for diabetes, we look at blood glucose, the HOMA index, which is calculated by looking at the insulin and glucose level divided by 22.5. And we also look at the hemoglobin A1C. When we look at cigarette smoking, we categorize cigarette smoking into 3 kinds of groups. We look at the current smokers, and those are defined as those who answered yes to the question, do you smoke cigarettes? And also they had a serum nicotine level more than 3.0. The second-hand smokers, which is what we are more interested in, are those who said no to the question, do you smoke cigarettes? And then they still had a nicotine level between 0.05 and 3.0. And these cutoffs were based on Dr. Benoit's study with his colleagues. So thank you very much. And the non-smokers were those who said no to the question due to smoke cigarettes and also had a less cotinine level of less than 0.05. There were other independent variables such as age, gender, race, physical activity, and also alcohol consumption. This analysis, we use the statistical analysis system with the software used to analyze data, especially complex survey design like the Enhance. And for our outcome variables, for BMI, we categorize using a cutoff of 25. For HOMA index, use a cutoff of 2.2. For fasting glucose, a cutoff of 126. And for HbA1c, a cutoff of 6.5. And these cutoffs, those that were less than these cutoffs were considered normal, and above were abnormal. We use a statistical approach called the generalized linear model to look at the adjusted means and the standard errors of our outcome variables, looking at HOMA, fasting blood glucose, hemoglobin A1c, and BMI, after controlling for these risk factors such as age, gender, race, physical activity, and alcohol consumption. We then conducted a logistic regression model controlling for just the demographic variables, that is age, gender, and race. So in our study sample, we can see that 34% of our participants were exposed to secondhand smoking. Majority of our study participants were non-Hispanic white, about 73%. Female were 51%, and 44% of our study sample said they had consumed 2 to 3 drinks per day in the past 30 days. And also 67% physical activity. These were those who said yes to engaging in moderate or vigorous activity over the past 30 days. If we are interested in secondhand smoke, we look at those who said no to the question is smoking or not, but then we look at those who had a cotinine level of 0.05. When you look at this graph, you can see that among non-Hispanic blacks, 55% said they do not smoke, yet there was a detectable amount of cotinine in their blood. Non-Hispanic whites were about 39%, and Hispanics were 44%. And this is the next slide showing that when we look at the 3 races, African Americans were the ones with the most exposed to secondhand smoke, even though they said they did not smoke cigarettes. Now, so this is the main point. So this is the adjusted mean and standard error of our outcome variables. For BMI, you can see that secondhand smokers were statistically significant from the non-smokers. The mean BMI was about 28 compared to the non-smokers, that was about 27. And here you can just compare that current smokers are statistically different from non-smokers, but they have a lesser BMI and are more leaner than non-smokers. When we look at the HOMA index, there was no difference between the current smokers and the non-smokers. However, when you look at the second-hand smokers, there is a statistically significant difference with the non-smokers. For the second-hand smokers, the HOMA index was 2.16, whereas for the non-smokers, it was only 1.99, showing that there is a difference between the second-hand smokers and the non-smokers, that the higher risk of having diabetes. When we look at the HbA1c, both the second-hand smokers, and the current smokers were statistically different from the non-smokers. The second-hand smokers and the current smokers had almost the same, had literally the same adjusted mean of HbA1c, so showing second-hand smoke is as bad as smoking. Looking at the fasting blood glucose, there was no statistical difference between the current smokers and the non-smokers, but there was a difference between the second-hand smokers and the non-smokers. The second-hand smokers had a fasting blood glucose of 99.87, almost 100, compared to non-smokers who had a fasting blood glucose of 97.82. And these data are all adjusted for the risk factors such as age, gender, race, alcohol, and physical activity. We then look at the adjusted odds ratio and the 95% confidence interval of our outcome variables. As I mentioned earlier, we looked at the cutoffs of BMI, fasting blood glucose, hemoglobin A1c, and the HOMA index. For BMI greater than or equal to 25, we can see that the current smokers are again leaner, they have a lower BMI than the non-smokers. Whereas the secondhand smokers have a higher BMI of about 1.2 compared to the non-smokers. For fasting blood glucose, we can see that both the secondhand smokers and the current smokers have a higher odds of being at risk for diabetes, having a higher fasting blood glucose compared to the non-smokers. For HbA1c, there was no difference with the current smokers. Looking at this graph right here, but for the secondhand smokers, they were about 1.4 times higher of having a higher HbA1c compared to the non-smokers. And when we look at HOMA, there was no difference with the current smokers, but again, secondhand smokers had a higher odds of having a higher HOMA, and that's having a higher risk of getting diabetes compared to the non-smokers. And this adjusted also was adjusted with demographics of age, gender, and race. And based on the results that I just discussed, we can conclude that secondhand smoking is associated with both obesity, overweight, and diabetes. And also, but we need to do more causal studies to show the relationship between secondhand smoke and obesity and diabetes to show the mechanisms involved in this association. Unfortunately, ENHANCE is a cross-sectional study, meaning there was no follow-up, so we couldn't ascertain a causal relationship to conclude that secondhand smoke causes diabetes or obesity, but we can just say there was a relationship and an association that we need to find more follow-up studies to conclude those results. And also, even though we control for factors such as age, gender, race, physical activity, and alcohol consumption, there might be some other confounders such as C-reactive protein or other biomarkers that we would look into to see if this relationship still exists. Based on the results that we got, we need to put more effort at reducing exposure to secondhand smoke, because from these results we can see that being exposed to secondhand smoke is as bad as smoking cigarettes. This study was funded by the MEDA, NIMHD, and thank you.
Great job.
Thank you.
Thank you. Questions?
I have a question. Oh, wait till we ask this.
Dr. Benowitz, then we'll be you.
Thank you.
I have two questions. One is, we're able to look at socioeconomic status as we know that people who have poor and have worse diet, that's the first question. Second of all, have you done a product in a mediation analysis to see if the diabetes parameters are all explained by BMI?
Yes. So that is something that, thanks for your question. You want to repeat the question?
Yes.
Oh, sorry, go ahead.
Yeah, go ahead. Go ahead.
So Dr. Benowitz wants to know if we look at different mediations with diabetes to see if there's so to look and see if we looked at other social economic factors such as diet or education. That's something that we are going to look into. We are writing a grant, and these are factors that we are going to get money and work on. Thank you.
Did you control for socioeconomic status?
No, we didn't.
Okay.
So that's something we're going to do.
A very good point.
Yes.
You might see that the secondhand smokers have lower socioeconomic
Right.
status, and that may be why they have more diabetes.
Exactly.
We have one from UTEP, then we'll get Dr. London. UTEP? No?
UTEP has a question.
Yeah, go ahead, UTEP.
Have you thought about the possibility that diabetic persons may have a different type of nicotine, and have you looked at that or thought about it, and what would you predict the codeine levels would look like following a smoking session?
There's a few more answers.
You know that better than I, Laura. Codeine is, the metabolism of codeine is crucial, and Dr. Benowitz, is also talking about that.
Oh, that's right.
Nicotine metabolism metabolism of protein is crucial. One of the interesting findings that Dulcie just presented today was about the African Americans having higher rates of second and active levels. Possibly one is socioeconomic status, and second of all, if they don't metabolize protein, they break down protein as much, so they're for a given amount of exposure, the protein levels can be higher. And then your second question, Laura, was on diabetes?
Yes.
You want to repeat that again?
What would you predict the protein levels would look like in your prediction of how diabetics would respond to a smoking session?
I don't know. I think you guys are looking at that, and we'll see. Great question. Dr. Wilson?
I'm sure you thought about doing the studies in animals to separate the effect of nicotine from other treatment in the very.
So, we others have looked at nicotine effects on diabetes in animals, and it's controversial, it's difficult. I think some of it's related to stress response. Most literature seems to think that nicotine has a reduction in diabetes in mice. The glucose tolerance test is less than control animals. We found variable data with that. Sometimes there might be more higher levels. And I think it also has to do with nicotine giving weight loss. As in the primary smokers here in the humans, the weight loss helps protect diabetes. In the animals, maybe that's enough to, they don't get diabetes. In the humans, it doesn't seem to protect it.
Thank you.
I mean, the data look really good, but and it's significant. But I don't know if the difference between 6.31 versus 6.38 in terms of this.
HbA1c. So, right, medically it might not look different, but statistically it is.
Because basically, it's to statistical analysis, but if that statistical analysis is really, I think that 6.31.
I think the answer to that question is a great question here. It has to do with odds ratio. So I think the odds ratios are probably more helpful to look for it. If you look at the odds ratio of greater than 50%,
Sorry.
5.5-fold in second-hand smoker. So that you have a 1.5 chance or 50% more chance of getting diabetes if you're a second-hand smoker. And there was 70 million second-hand smokers in the United States based on the population. We're talking about an extra maybe 30 million people in the United States might get diabetes. It's definitely whole. So even though we're a population, it's a very small percentage. We add that up with millions of people in the United States exposed to second-hand smoke. I think it makes a big difference.
It's not a question, this is a comment. This should not be considered a study. This should be classified as drug-induced diabetes.
Drug-induced diabetes.
Okay.
All right.
I think we got to move on to the talk. Sorry. Okay.
Since the non-smoking is a problem and it doesn't have effect on the non-smokers, what are your steps and the future plans to curb the
Great question. So the question, how can you reduce secondhand smoking? Elsie, do you want to make a comment on that, or?
I believe that will go with Dr. Benewitz's plan and what they are doing, policy changes. And I think personally, try and avoid being around smokers, personally.
Right. So I think, this is one of the messages we want to get out of our study that people that say, secondhand smoking is benign, I'm smoking in my backyard, so what's it, my neighbor is against it, or I'm smoking next to my colleagues at work, I'm at the casino, or the old days on the airplane, you're saying fine. Well, our message is not benign, that's what Dr. Benewitz's message is. So we have to have policy in place to prevent it. And California is the forefront of the state that has policy against smoking in public places. Many states in the Midwest and South don't have those policies yet. So I think we want to go and help us get these policies across the country. Thank you.